Physiology of renal system. This is a series of videos on the renal system physiology. This would be an introductory video to the renal system. In this video, we will talk in general about the kidneys, the renal blood flow, and at the end of the video, we will take a look into a general functions of the renal system. Like and subscribe for more free medical contents. The renal system is composed of 2 kidneys, 2 ureters, the urinary bladder, and the urethra. Most of the topics in this series will be about the kidneys. We have a video on the micturition reflex related to the urinary bladder and the urethra, but most of our videos will be on the kidneys. Kidneys are bean-shaped retroperitoneal structures located at T12 to L3, protected by the 11th and the 12th ribs. The right kidney is situated just below the liver, while the left kidney is situated below the diaphragm. The right kidney is pushed down by the liver, and that's why it's slightly lower than the left kidney. The kidney is composed of outer renal capsule, which is a fibrous connective tissue protecting the kidneys from the outside. We find the renal cortex when we remove the renal capsule, and the cortex is the outer layer of the kidney. Deep into the cortex, we find the medulla, the inner layer of the kidneys, composed of about 10 renal pyramids. Renal lobe is a section composed of part of the cortex and 1 renal pyramid from the medulla. Medullary pyramids empties its contents into the minor calyx. Minor calyx empties into major calyx. All major calyces empties into the renal pelvis. Then the contents pass to the ureters and then into the urinary bladder as urine. Renal blood flow. As we mentioned before, the kidneys are bean-shaped. It has a concave structure, and on the concavity of it we find a structure we call the renal hilum. The renal hilum is where blood vessels and nerves supplying the kidneys enter and leave, and we also find the ureters exiting through the renal hilum. Renal blood flow is about 1 to 2 liters per minute. It's about 20 to 25% of the cardiac output. 90% of the renal blood flow supply the renal cortex, while only 10% supply the medulla. Regulation of renal blood flow. The renal blood flow is characterized by something we call autoregulation, meaning that the kidneys could control the amount of blood entering, and thus it's not affected by changes in systemic blood pressure. The autoregulation process is explained by myogenic response, which is the smooth muscles of the kidney arterioles. It constricts and dilates, adjusting the amount of blood entering the kidneys. Also, we have humoral factors, and these are the substances that are mixed with blood, and it exerts its effects when reaching the kidneys. For example, nitric oxide causes vasodilatation. Angiotensin II is a strong vasoconstrictor. Other causes: the sympathetic nervous system. It decreases the renal blood flow by causing the renal arterioles to constrict. Catecholamines decreases the renal blood flow by vasoconstriction. Dopamine increases renal blood flow by vasodilatation. Prostaglandins. Some of these prostaglandins are dilators, like prostaglandin I2, and some of them are constrictors, like thromboxane A2. And that's why chronic use of aspirin decreases renal blood flow and may contribute to acute renal injury. Aspirin inhibits the synthesis of prostaglandins that are important for vasodilation, thus leading to a lower renal blood flow. And the final segment of this video about general functions of the renal system: excretion of waste products. The kidneys get rid of toxic substances of the body, like urea and uric acid, by excreting them into urine. Regulation of extracellular fluid volume. And the kidneys regulate the extracellular fluid volume by either excretion of excess water into urine or preservation of water. If someone is dehydrated, for example, the kidneys reabsorb water back into the body, thus increasing the extracellular fluid volume. Regulation of extracellular fluid osmolarity. by reabsorption or excretion of solids, thus regulating osmolarity. Regulation of extracellular fluid electrolytes by excretion and reabsorption of solids. Regulation of systemic blood pressure. The kidney cells are sensitive to pressure changes. Some kidney cells produce an enzyme we call renin, which is crucial for blood pressure control. pH regulation, and the kidneys regulate the acid-base balance, and we're going to have a separate video about the acid-base balance. And last but not least, the endocrine functions of the kidneys. The kidneys produces hormones like erythropoietin, which is an important hormone for the red blood cell synthesis. And that's why people with chronic kidney disease often suffers from anemias. Vitamin D activation takes place in the kidneys. And as we mentioned before, renin is produced from kidney cells, and it's so important in blood pressure regulation. So in this short video, we're going to look into the kidney cells. It's called the nephrons, and it's the basic functional unit of the kidneys. There are about 1.2 million nephrons on each kidney on average. The nephron is composed of two parts, the glomerulus, and the glomerulus does something we call filtration. It turns blood flowing to it into a pre-urine, or we call it a filtrate. Second part of the nephron is the tubular part. After the filtrate or pre-urine passed by the glomerulus reaches the tubules further adjust the fluid by two processes. We call them reabsorption and secretion. By preserving molecules that the body needs and getting rid of excess or toxic molecules that the body wants to get rid of. So according to the body needs, reabsorption and secretion takes place on the tubules. Tubules are composed of the proximal convoluted tubules, continuous with the glomerulus, loop of Henle. It has a descending and an ascending part. The ascending part of the loop of Henle has a thin part and a thick part. The thick part of the loop of Henle is continuous with the distal convoluted tubules. Distal convoluted tubules of many nephrons then empty its contents into the collecting ducts. The collecting ducts excretes urine into the minor calyx, the major calyx, and renal pelvis, as explained in our previous video. Urine reaches the ureters and then excreted from the body as urine. Our previous video was about the general components of the nephron. In this video we get into more details about the glomerulus. We will be looking into the cell types and the urine formation process. Glomerulus is a tuft of capillaries. Afferent arteriole enters the glomerulus, forming these capillaries, which drains into efferent arteriole. So afferent arteriole entering and efferent arteriole leaving the glomerulus. These capillaries we call the glomerulus are enclosed within the Bowman's capsule. The Bowman's capsule is composed of three layers: an outer parietal layer made of simple squamous cells and an inner visceral layer. The inner layer is made of specialized cells we call the podocytes. Podocytes have a foot-like projections called pedicles. Many food processed from multiple podocytes form something we call filtration slits. Filtration slits help in the process of glomerular filtration. It's one of the three layers of the glomerular filtration membrane. Third layer of the Bowman's capsule is the capsular layer. It's the layer in between the parietal and the visceral layer. It's a space where filtrate or pre-urine enters from the glomerular capillaries. Now let's talk about the glomerulus. As we mentioned before, it's the capillaries enclosed within the Bowman's capsule, supplied by afferent arteriole, drained by efferent arteriole. There is a specialized types of cells on the afferent arteriole called the granular cells or the juxtaglomerular cells. Granular cells are sensitive to changes in blood pressure. It has mechanoreceptors on its surface. Granular cells reacts to low blood pressure and sympathetic stimulation. There is also a third factor which we're going to discuss on our next video about the juxtaglomerular apparatus Comment below if you know the third factor stimulating the granular cells. So in response to low blood pressure or sympathetic stimulation, granular cells synthesize, store and secrete an enzyme called renin. Renin is an enzyme that works by converting a protein that is synthesized from the liver called angiotensinogen into angiotensin 1. Angiotensin 1 is converted to angiotensin 2 by an enzyme found in the lungs called angiotensin converting enzyme. Angiotensin 2 is a potent vasoconstrictor that raises blood pressure to correct the hypotension. It's part of the renin-angiotensin aldosterone system, and we're gonna have a separate video for that topic. Now, with a small clinical note, when a patient has a renal artery ischemia from thrombosis, for example, juxtaglomerular cells are hyperstimulated by the lower blood flow, and they release too much renin into the blood, leading to very high blood pressure. Other specialized types of cells we find around the glomerular capillaries are the mesangial cells. Mesangial cells form structural support for the glomerular capillaries. They also have the ability to engulf foreign or dead tissue by phagocytosis. Mesangial cells are contractile cells. They have actin and myosin filaments. It does not have mechanoreceptors like juxtaglomerular cells, but it is sensitive to changes in tension or pressure on the glomerular capillaries, and they contract or relax, adjusting the surface area and thus helping in the regulation of renal blood flow. Our previous video was about the glomerulus. In this video, we will continue on other parts of the nephron. First, we start with the proximal convoluted tubule. This is where reabsorption begins. Next comes the loop of Henle, which has two parts, a descending loop of Henle and an ascending loop of Henle. The ascending limb has a thin part composed of flat cells and a thick part made of cuboidal cells. After the loop of Henle, the filtrate enters the distal convoluted tubules. This part contains a specialized types of cells called the macula densa. These macula densa cells are chemoreceptors. They detect the levels of sodium and chloride in the fluid. If the sodium level is too low, they send signals. These signals go to the juxtaglomerular cells, which are located in the wall of the afferent arteriole. In response, the juxtaglomerular cells release an enzyme called renin. So what makes the juxtaglomerular cells release renin? There are three main triggers. First, the low blood pressure, and second, the sympathetic nervous system stimulation as mentioned in our previous video. The third factor that stimulates the juxtaglomerular cells to release the renin is low sodium levels detected by the macula densa. These cells, the macula densa, the granular cells, and another type called RAAS cells, together form the juxtaglomerular apparatus. Let's look at the nephron structure in real anatomy. The distal convoluted tubules comes back near the afferent and efferent arteriole, close to the glomerulus. In this area, the juxtaglomerular apparatus is formed. Now let's break down the three types of cells in the juxtaglomerular apparatus. First, the juxtaglomerular cells or the granular cells found in the afferent arterioles, and these cells release renin. Second, the macula densa cells found in the distal convoluted tubules. They sense the sodium levels and send signals. And third, the RAAS cells or the extraglomerular mesangial cells, and they may help in transmitting signals, but their function is still unclear. So do not confuse them with the regular mesangial cells, which help with support, contraction, and phagocytosis as mentioned in our previous video. Those three types of cells together are called the juxtaglomerular apparatus. Now we come to the collecting ducts, the final part of the nephron. It contains two types of cells. The principal cells, they make up about 75% of the cells of the collecting ducts. These cells have receptors for aldosterone and ADH. They help control water and salt balance. Then we have the intercalated cells, and these help with the acid-base balance, meaning they help with controlling the body's pH. We will explain these cells in more details in a separate video. In this video, we are diving into the blood supply of the kidney and the two main types of nephrons you must understand. Remember, the kidney has two main regions: the cortex, which is the outer layer, and the medulla, the inner core made up of pyramids. As mentioned in our previous videos, each renal lobe includes a medullary pyramid plus the cortex above it. And within that, we have renal lobules, which is a cluster of nephrons draining into one collecting duct. Now let's zoom in on how blood flows through all of this. The kidney's blood supply begins with the renal artery, which branches straight off the abdominal aorta, a big highway of blood. This renal artery splits into segmental arteries, the main branches that actually enters the kidney. Next stop, the interlobar arteries. They travel between the medullary pyramids, heading towards the border between the cortex and the medulla. There, they curve around and become the arcuate arteries, like little arches around the top of the pyramids. Then come the interlobular arteries, shooting up into the cortex. From these, tiny afferent arterioles branch off, and those are vessels that feed into the glomerulus of each nephron. Now here is where it gets really interesting. After blood is filtered through the glomerulus, it exits through the efferent arteriole, and what happens next depends on the type of the nephron. In cortical nephrons, the efferent arteriole gives rise to the peritubular capillaries. These capillaries wrap around the tubules and play a key role in tubular reabsorption, where filtrated substances like glucose, water, and electrolytes return back to the bloodstream, and tubular secretion, where certain waste products are added to the tubule from the blood. But in juxtamedullary nephrons, things go deeper, literally. The efferent arteriole forms long straight capillaries called the vasa recta, which dive deep into the medulla. These vessels are crucial for concentrating urine. After all this, the blood begins its return journey through interlobular veins to arcuate veins to interlobar veins, then into segmental veins, and finally the renal vein, which exits the kidney and rejoins circulation through the inferior vena cava. Alright, now let's break down the two types of nephrons. Cortical nephrons, and they make up about 85% of all nephrons, located in the outer cortex, and they have short loop of Henle. Cortical nephrons are surrounded by peritubular capillaries, and they are more involved in basic filtration and reabsorption. The other type is the juxtamedullary nephrons, and the juxtamedullary nephrons make about 15% of the total number of nephrons found at the junction between the cortex and the medulla. They have long loops of Henle that go deep into the medulla. Juxtamedullary nephrons are surrounded by vasa recta, and they are essential for urine concentration. In this video, we are diving into the first step of urine formation, glomerular filtration. Remember, urine formation has three main steps: filtration at the glomerulus, then reabsorption and secretion in the tubules, and finally excretion of what's left. Today we are zooming in on filtration, how blood enters the kidneys, passes through specialized capillaries, and gets transformed into something we call filtrate. So let's begin. Blood enters through the afferent arteriole and reaches a tight ball of capillaries called the glomerulus. Here is where the magic happens. Some substances in the blood get filtered through a specialized barrier and enter Bowman's capsule. The resulting fluid is called filtrate, or you could say pre-urine. But not everything gets filtered. Larger components like blood cells and certain proteins exit through the efferent arteriole instead. Let's now explore the star of this process, the glomerular filtration membrane. This membrane has 3 key layers, and each one plays a special role. First, the endothelial cells of the glomerular capillaries, the basement membrane, and the third layer is the visceral layer of the Bowman's capsule, the podocytes. So let's get into more details on each one of them. The first layer is the endothelium of the glomerular capillaries. It's full of small pores called fenestrations, about 70 nanometers wide. These allow tiny molecules like water, glucose and ions like sodium and chloride to pass through. But larger things like red blood cells, white blood cells and platelets, nope, they are too big to pass through this layer. The second layer is the basement membrane, and it's not just a structural layer, it has a negative charge. This acts as filter by size and charge. So if a molecule is too large or negatively charged, it's blocked. For example, the protein albumin has a size of 69 kilodaltons and a negative charge. So sorry albumin, you are not getting through. The third layer is formed by podocytes, the visceral layer of Bowman's capsule. These podocytes have food processes that interlock, forming small slits called filtration slits. Between the slits and the negative charge, only certain substances can pass through. Let's go through a few of them. Small molecules like water, 0.3 nanometer. Sodium ions, 0.9 nanometer, and chloride ions can pass through easily. Even though chloride is negatively charged, it's so small it sneaks by. Same for glucose, 180 daltons, urea 60 daltons, and creatinine about 113 daltons. All small, all able to filter through the membrane smoothly. Now let's talk about what cannot pass through. Blood cells, way too large, measuring over 6 to 8 micrometers. Plasma proteins like albumin, large and negatively charged. Protein-bound substances like calcium bound to albumin. Even free hemoglobin due to size and charge cannot make it through. So why is this important clinically? If we find red blood cells or proteins in a patient's urine, that's a red flag. It likely means there is a damage to the filtration membrane. For instance, in glomerulonephritis, you will see red blood cells in urine. In nephrotic syndrome, you will find significant protein loss. We call it proteinuria. And that's a wrap on the glomerular filtration membrane. We've seen how your kidney acts like a fine-tuned filter, letting the good pass and blocking the rest. If you enjoyed this breakdown, give it a like and subscribe for the next video in our urine formation journey. We are heading to factors affecting glomerular filtration membrane. Stay curious, future doctors, and I'll see you there.